Hello everyone, I am going to speak about oncogenic retroviruses. Definition of oncogenic retroviruses. An oncogenic retrovirus is a virus that can cause cancer by integrating its genetic material into the host cell's DNA, leading to uncontrolled cell growth. Oncogenic retroviruses are RNA viruses. They contain a single stranded RNA genome. Those virus causes a wide range of tumors but are most commonly associated with either tumors involving hematopoietic cells. And hematopoietic cells is an miniature cell that can develop into all types of blood cells including white blood cells and red blood cells. Examples of oncogenic retroviruses include the human T cells leukemia virus and certain strains of the feline leukemia viruses. Morphology. They are enveloped virus with a spherical or pleomorphic shape. Pleomorphic shape is a non-genetically encoded variable particle shapes enable pleomorphic virus to overcome selective pressure. And under electron microscopy, retroviruses topically exhibit a core structure surrounded by a envelope. The core contains the viral RNA genome along with enzymes necessary for viral replication such as reverse transcriptase, integrase and protease. Inside the viral core, the RNA genome of oncogenic retroviruses is single-stranded and positive sense. Epidemiology, geographical distribution. The distribution of oncogenic viruses may vary geographical. For example, human T cells leukemia virus type 1 is more prevalent in certain regions such as Japan, the Caribbean, part of Africa and South America. Geographical distribution is here the natural arrangement of the virus from of animals and plants in the different region and locate of the earth. Transmission routes. Oncogenic retroviruses can be transmitted through various routes including sexual contact, blood transfusions, and from mother to childbirth during childbirth or breastfeeding. Risk factor for infection may include unprotected sexual activity, sharing needles among injecting drug user and receiving blood transfusion from infected donors. Population groups. Certain population group may have a higher prevalence of infection with oncogenic retrovirus. Incidence and prevalence. The incidence and prevalence of cancers associated with oncogenic retrovirus may vary over time and across populations. Factors such as changes in behaviors, improvements in screening and diagnostic method and healthcare interventions can influence those trends. Impact on health. Oncogenic retrovirus can have a significant impact on public health, particularly in regions where they are endemic. Endemic is a regularly acquiring within an area or community. When it is present but limited to a particular region, that is a endemic. They can contribute to the burden of certain types of cancer such as adult T cell leukemia. T cells leukemia virus is a rare and aggressive peripheral T cells neoplasm associated with human T cells leukemia virus. Lymphoma. Lymphoma is a broad term for cancer that begins in cell of the lymphatic system caused by and certain types of leukemia and lymphoma in animals caused by retrovirus such as cats. Transmission of oncogenic retrovirus. Oncogenic retrovirus can be transmitted horizontal through direct contact such as sexual intercourse or blood transfusion or vertically from mother to child during pregnancy. childbirth, or breastfeeding. Horizontal transmission is more common among humans, while vertical transmission is often observed in animals. Next, incubation period. Incubation period, the time period between exposed to a pathogens and the first symptoms appearing in the take time in the host cells. The incubation period for oncogenic retrovirus can vary depending on factors such as the specific virus, host immune response, and individual characteristics. In some cases, symptoms may develop relatively quickly, while in others, it can take months or even years for clinical manifestation to appear. The clinical manifestations variable observable symptoms used in diagnosing disease. Next, risk factor exposed to infected body fluid. Exposed to infected body fluids is a occupational exposed to blood and body fluids is a serious concern for health care workers, presents a major risk factor for cancers in transmission of infection diseases such as example hepatitis B and hepatitis C, HIV, human immunodeficiency viruses. Immunosuppressive patient having a weakened immune systems. Vertical transmission. Vertical transmission is a example COVID-19 from mother to child was not possible. Pathogenesis entry into the host cells. Oncogenic retrovirus such as human T cells, leukemia virus, and human immunodeficiency virus enter host cell through interactions between viral envelope proteins and host cells receptors. This allows the virus to gain entry and begin replicating within the host cells. Entry into the host cells viral envelope proteins is a membrane protein. The envelope consists of an inner layer of lipids. Integration into the host genome, the DNA drying the cell divisions. So drying a mitotic cell cycle, the DNA contains per chromosome doubles, drying space each chromosome strains as the all chromosomes start as one chromatide. On the host cells, the retroviral RNA genome is reverse transcribed into the DNA by the viral enzyme reverse transcriptase. The resulting DNA molecules known as a previous ingredient in the host cell's gene. This ingredient is a step in the pathogenesis of oncogenic retroviruses because it allows the virus to persist within the host cells and replicate along with the host's DNA during cell divisions. Activation of oncogenes. The integrate previous may disturb normal host cell gene expression, leading to dysregulations of cellular processes such as cell growth, differentiations, and apoptosis. Retroviruses carry genes called oncogenes which can promote uncontrolled cell proliferations and tumor regenesis when activated. Immune evasions. The most the most of the host mRNA, the formation of replication, original stress, granules formations, membranes, modifications, actions of protease. That is immune evasions. Immune evasions oncogenes retrovirus have enveloped various mechanisms to evidence host immune response, allowing them to establish persistent infections and promote tumor regenesis. Chronic inflammation, failure of eliminating the agent causing an acute inflammation such as infections organisms including oncogenic virus. Infection with oncogenic retrovirus can induce chronic inflammation which creates a microenvironment conducive to tumor growth and progressiveness. Replication, attachment and entry. The virus attaches to receptors on the host cell's surface and enters in the cells. Reverse transcriptase. The viral RNA gene is reverse transcripted into the DNA by the enzymes reverse transcriptase. Integration. The newly synthesized viral DNA is integrated into the host cell's genome, become a provirus. Transcription and translocation. The proviral DNA is transcribed into the RNA and translocated into viral proteins by the host cell's machinery. Assembly and budding. Assembly. Viral RNA and proteins are assembled into new viral particles at the host cell's membranes. Budding and release new viral particles and word of form the host cells ready to infect other cells. Throughout this process in integrate of the viral DNA into the host gene is a critical step that can lead to oncogenesis by disrupting normal cellular genes regulatory elements. Symptoms of oncogenic retrovirus: fever, fatigue, mouth rash, sore throat, swelling lymph nodes and muscle ulcers, neurological disorders, lymphadenopathy. Laboratory diagnosis: serological assay, ELISA, molecular assay, PCR polymer chain reaction, retroviral DNA or RNA sequencing in blood, histopathology, RT-PCR. Treatment of oncogenic retrovirus. Some therapy can treat in oncogenic retrovirus: antiretroviral therapy, interferon therapy, chemotherapy, targeted therapy, immunomodulatory therapy. Prevention and control. Education and awareness, need exchange programs, safe sex practice, blood safety measures, proper vaccination, good personal hygiene, avoid infected person. Thank you.